Good morning, everyone. Thanks for inviting me for this presentation. Today, I'm going to talk about developing a new pipeline for exploring pleiotropy of GWAS data at gene level. So, as a brief explanation about the context, when we talk about the pleiotropy, we mean that we're looking for shared variants or genes or pathways that can affect multiple traits at the same time. And it's important because studying pleiotropy can inform more about the common biological mechanism and also can be helped to disentangle relationship between phenotypes uncovered by epidemiological studies. And when we work with the GWAS data, most of the time we are not allowed to use the individual level data due to the policies of security. So most of the time we confront with GWAS summary statistics in, for example, 2 traits. And for each trait, we have the effect value and the p-value for that effect. And in the traditional way, we are looking for finding pleiotropy at SNP level. And then after finding the candidate SNPs, we go to the functional annotation of the SNPs. That is the most problematic step for interpretation. So one of the solutions that they suggest is that to analysis at gene level or pathway level in order to just interpret easier than SNP level and potentially would be more powerful. So to do that, we developed first a package called GCP base, which was based on another method called CP base. It was a Bayesian statistical methods that is based on the spike and slab players for variable selection. It uses Markov chain Monte Carlo methods through the empirical Bayes Gibbs sampler and try to just pleiotropy association in the SNP level only based on the summary level data. So we first extended the CP base method to detect pleiotropy also in the group level. Here means, for example, gene or pathway levels. And as a very skipping schema, I just talk about the statistical methods that we used. We have 2 different GWAS summary statistics data, for example. And then first we just test the H0 assumption. If the H0 assumption, which is the no association between the SNPs at trait X and trait Y, is not rejected, it means that there's no signal and no variable pleiotropy, and also there's no group pleiotropy. But if this test was rejected, we compute a value called theta. Theta is the probability of at least 2 traits have non-zero effects in the condition of having the data. So after computing theta, if the theta exceed the threshold value, you can say that we have the candidate gene that might have the pleiotropy effect. And because it's in the probability status that we're talking about, we consider 0.5 as the threshold, but the user can use any threshold that wants. But the problem is when you work with genome scale GWAS data, the problem is there are lots of things you need to do pre-processing before going to the pleiotropy finding process. So we just provide a user-friendly pipeline that do all the analysis from the scratch, from the start to the end to the user, and then we apply this pipeline to the example of the breast and ovarian cancer GWAS summary statistic data. This is the overview of the pipeline that we developed. It has 3 main steps and also 1 optional steps. At first, it's consider 2 GWAS summary statistic data, try to just do the harmonization between 2 data. You know that if we need to just be careful about the effect allele and non-effect allele in 2 different data sets. So we first need to harmonize these data. If it's not the same, we need to just change the sign of the beta or effect. So we do this through this step. And then if a user provide the annotation files, but we provide the annotation files by default. But if a user has another annotation files, it can use the annotation files to just annotate SNPs to the genes. And then if the number of SNPs that are located to a specific gene is high, that reduce the running time, the user have this option to use LD clumping steps in order to shrink the number of SNPs. And after that, the input is ready to run through the GCP-based package without any further changes. And the output would be the candidate list of genes or pathways that candidate genes here, because we just use the annotation of the genes. And in each step, there are different visualization steps that gives the hint to the user that's what can do in further analysis. For running the pipeline, at the moment, there are 3 options for running the pipeline. One is using the bash files that do everything automatically. And also we provide the R file that do this the same for the one that are more familiar with R than the bash. And also we have designed the Shiny app for the one that are not familiar with, they are scared of working with command line, so they can use the Shiny app and provide everything in graphical view. And then just by clicking different options, they can run the pipeline. For application, we use the common, we use the 2 consortium data for BCAC and OCAC for breast and ovarian cancer. The number of SNPs are about 10 million for breast and 18 million for ovarian. And the number of coding genes was about 18,000. That's ranged from 188 to more than 1 million base pair. If we go through just the SNP level, so we can see that there are 3 common significant signals in the SNP level at chromosome 5, 10, and 15. But when we go through the gene level analysis, you can see that there are the number of pleiotropic genes with theta greater than 0.5 is 151 located in 79 genomic loci. This is the number of genes, and for better understanding, we just provide the locus of that genes that they are located. The 3 green locus are the one that we showed in the GWAS, and it's also appear here. The red one was the locus that the genes that located there has been previously mentioned in the literature, and also the black one are the one that are assumed as a new potential pleiotropic genes. And if you go through the visualization steps, you can see that at a glance you can find out which are the distribution of the pleiotropic genes in around the chromosomes, and also the karyotype plots, and also the circles plots for having the overview of the distribution of the pleiotropic genes, and the number of distribution of the theta value, and also the number of SNPs for the distribution of number of SNPs for every gene. And as always, as a running time, if we run this analysis on a Intel Core i7 computer with 32 gigabyte of RAM, the reformatting and annotation steps took about 30 minutes. And if you run all the analysis in one CPU, it takes about 16 days to just finish the running. But the pipeline has this ability to just mention the number of CPUs. And if you use 6 CPUs at your laptop, for example, it reduces the number of running time drastically. So, and if you use more CPUs, you can also have less time. And for your information, the very detail of every steps is available in the GitHub page one by one. And we have also publish the package and the pipeline. And at the end, I need to thank all our collaborative members of the groups. And I'm happy to answer all your questions. Thank you.
Many thanks. We have time for a quick question. Anyone? Okay.
Hello, thank you for your presentation. I actually have 3 quick questions. The first one is if I can use more than 2 GWAS as inputs, because in your examples you use it always 2 summary statistics, but I could, for example, use more than 2 summary statistics file as inputs.
Yeah.
And if I can mix the GWAS that was performed in 2 different populations, for example, European populations and African populations.
Yeah, but at the moment that the pipeline doesn't have such options, but it's an option that we can just add it, but you can do it by yourself and then put the final results as an input. Okay, thank you.